November 24, variant 2. Photosynthesis. The experiment is about photosynthesis. It occurs in chloroplasts. Students investigated the effect of light intensity on the rate of photosynthesis. I'm going to change the light intensity and I'm going to measure the rate of photosynthesis. Okay, so the light intensity is my independent variable. How am I going to measure the rate of photosynthesis? He gave me a new way. Blue DCPIP. Blue DCPIP can be used to investigate photosynthesis. We took the DCPIP before as a test for vitamin C. In this experiment, he gave you something else. I can use it to measure the rate of photosynthesis. During photosynthesis, the dark blue color of the DCPIP changes and becomes colorless. So if the photosynthesis is fast, it will take less time to change colors. If the photosynthesis is slow, it will take more time. A chloroplast suspension that is made from spinach leaves has green color. Green color. Prepare a chloroplast suspension from the leaves, spinach leaves. Label one test tube A and another one B. Put 1 cubic centimeter of the chloroplast solution in A and B, both of them the same volume. So this is a controlled variable. Put A and B into ice cold water bath for 2 minutes. This is also a controlled variable. And then 5 cubic centimeter of DCPIP into test tube A. Put it in the rack 10 centimeter from bright light. Okay? Turn on the light, start the stop clock. What am I going to measure بقى؟ يبقى I worked on test tube A first. I put the chloroplast suspension and the DCPIP, and then I put it 10 centimeter away from the light. Start the stop clock. Measure the time taken for the blue liquid to return to green. Why is it going to return to green? It will be colorless. When it is colorless, the color of the chloroplast suspension will show. So it will be green. Reset the stop clock to 0. Move the test tube rack so it is 30 centimeter from the bright light. And then I will do test tube B back. Chloroplast suspension, DCPIP, I will put it in the rack, which is 30 centimeter away instead of 10 centimeter. Put the test tube in the rack, start the stop clock, measure the time taken to turn to green color. And these are the results in minutes and seconds. What does he want you to do? Prepare a table, record the results, convert the times to seconds. Okay. What did I change? Changed variable is always on the left side of the table. I changed the distance from the lab. 10 centimeter for test tube A, 30 centimeter for test tube B. What did you measure? I measured the time to change to colorless. And then I will convert it to seconds. Convert it to seconds. 1 minute 37 seconds يعني 60 plus 37 هنا 5 times 60 plus 43. I will calculate it. This is 397 and this is 343. This is my table. 4 marks for this table. What is the conclusion? Am I going to describe the results? The more the distance, the more the time? No, I'm going to tell him a conclusion. As the light intensity increases, the rate of photosynthesis increases. When you put the plant closer to the lamp, this means you have high light intensity. So it took less time to change colorless, which means that the photosynthesis is faster. Photosynthesis is faster at higher light intensity. Okay? Dependent variable. Dependent is the results. What are the results that I was measuring? I was measuring the time for color change. This is the dependent. What are the possible sources of errors in step 7 and step 11? Let's see what's step 7 and step 11. Step 7, measure the time taken for the blue to return to colorless, return to green, sorry. Any experiment that has changing point for the color, what error do I say? It is difficult to judge the end point. My eyes cannot determine in which second exactly did this happen. So the error will be difficult to judge the end point. What is the improvement? I should use a colorimeter, which gives me a number. When the second it's colored, so it will give me a number. It's a better way. DCPIP is used in food testing. Which substance? We just said it. DCPIP is used to test for vitamin C. Vitamin C. Figure 1.2 is a photograph of a seed from a dandelion plant. Dandelion plant. Okay. Draw it. I will draw the outline. If you have a zigzag outline like here, zigzag, and then straight, zigzag, and then a stalk, very thin, and then hairs at the top. Okay? Uh, this is how it should be. After the drawing, measure the line PQ. Measure the line PQ using your ruler, convert it to millimeter, calculate the actual length. Image over magnification, 19 over the magnification, which is 15 over 15. This is the actual length, 1.3 millimeter. Two significant figures. He got me another seed and he wants me to compare what is the similarity. Both have hair-like structures. What are the differences? This has a stalk in the middle. Here there is no stalk. Both have seeds that are longer than white. This is the similarity, longer than white. In عندي stalk هنا لأ. Here there is more hairs مثلا. I have to write which one am I talking about. Milk thistle is darker in color. The seed. He asked you not to include any references in size. But I cannot say this is smaller, this is bigger. I have to choose something else. Okay. Dandelion roots are used to flavor drinks. Students tested a sample of a colorless liquid for starch and reducing sugar. They found that it did not contain starch, it contains reducing sugar. So, what is the color of the iodine? If there is no starch, the color of the iodine will be yellow brown. How can I test for reducing sugar? Add Benedict reagent, heat in a water bath. If there is sugar, it will be orange red. Reducing sugar test in every exam. Okay, a new experiment, a new idea. Some types of weed killers prevent seeds from germinating. Plan an investigation to determine ايه بقى effect of weed killer concentration. Weed killer concentration. So I'm going to change the concentration of weed killer. This is my change variable. And what are the results بقى? Germination of seeds. How many seeds germinated? Uh, let's see the steps. Number one, replica. I will get 5 petri dishes to put the seeds in, and then each dish مثلا will have 10 seeds, 10 seeds in each dish on a wet cotton wool for the germination. What are you going to change? Weed killer concentration, different concentration in each dish. What are you going to put the same temperature, same type of seeds, same oxygen concentration, anything that affects the same volume of the weed killer. I only change the concentration. I will put everything that affects the germination the same. How are you going to measure your results? Check بقى how many seeds germinated out of the 10 seeds and calculate the percentage. Count the number of seeds germinated after a specific period of time. The last 3 points: safety precaution, improvement, repeat. Wear goggles so that the weed killer does not مثلا spray into my eyes. Repeat at least 2 times and calculate the average. I can also maintain the temperature around them using a heater, for example, to aid the room temperature, so it does not affect the results. Question 2. Investigated the effect of carbon dioxide concentration on the rate of photosynthesis. I'm going to change the carbon dioxide concentration and check the rate of photosynthesis in leaf disc. As photosynthesis takes place, it will give out oxygen bubbles. The oxygen bubbles causes the leaf discs to start to float. Sodium hydrogen carbonate is used as a source of carbon dioxide. Okay, take 5 leaves from the same plant. Use a cork borer to cut 50 leaf discs. Put 10 leaf discs into a syringe that has 10 cubic centimeter of sodium hydrogen carbonate solution. Remove the air from the air spaces in the leaf so that the leaf discs sink to the bottom. All the leaf discs will sink to the bottom. Position the syringe 5 centimeter from the lamp. When they photosynthesize, they will produce oxygen and they will start to float. Measure the time taken for at least 5 of the 10 discs to float to the top of the syringe. When there's 5 discs at the top, I will measure the time taken. Repeat the method with different concentrations of sodium hydrogen carbonate. And I want to change the carbon dioxide concentration. I will change the concentration of sodium hydrogen carbonate. Okay. The apparatus is shown. Draw and label one additional piece that could be used to reduce the heating effect. The lamp gives out light, who gives out heat. I put the heat filter or heat shield. The heat filter or the heat shield absorbs the heat. Only the light will pass. Say two variables apart from the temperature that were kept constant: volume of the hydrogen carbonate, the distance from the lamp, the type of the leaves, same plant, type of the plant. What is the independent variable? What did I change? What did I change in this experiment? I wanted to see the effect of carbon dioxide concentration. So this is what I changed. Carbon dioxide concentration. These are the results. Left side of the table on the x-axis. This is the x, this is the y. Right side on the y-axis. Put the labels, the x-axis, the y-axis. Put the scale, 0.2, 0.4, 0.6, 0.8. Here I have from 7 to 20, and from 0 to 20, 0, 5, 10, 15, 20 scale. Since he said plot a line graph, we always put the points and then join point to point using the ruler. The only case when we did it freehand was when he said a curve of best fit, our best fit line that looks like a curve. This is the only time that we need to use our hands. Okay. Estimate the sodium hydrogen carbonate concentration at which it took 12 minutes. 12 minutes. This is 11, 12 here. And then I will go down, check my number, put it here. For each of you, it might be a different number. If it is the correct reading from your graph, then it is correct. Calculate the percentage change in the time taken for the leaf disc to float from 0.2 to 0.4. 0.2, it was 20. 0.4, it was 17. It decreased. 17 final minus original over original times 100. Final minus original over original times 100. This is my number. This is the percentage change. Okay?